Welcome to the newest version of the JNM podcast. My name is Ken Herrmann. I'm a nuclear medicine physician based in Essen, Germany. And actually, we're going to do a big experiment this time. We're going to talk about the highlights of the Journal of Nuclear Medicine of 2025, what I like to call the All-Stars of the JNM. And to do this, I have three outstanding guests today who are going to discuss with me what has happened in the year 2025. And the first guest is actually someone who is coming back to the podcast. It's Professor Irene Buvat. She's a PhD professor of physics and also director of the research unit Laboratory of Translational Imaging and Oncology in Institut Curie in Paris, France. Welcome back, Irene.
Welcome, Ken, and thanks a lot for your kind introduction and for having me here today.
I'm also very happy that we have someone joining us at 6 a.m. in the morning, someone who has been before at the JNM podcast, Professor Thomas Roop. He's the Vice Chair of Clinical Operations and Strategy in the Department of Radiology, and he also serves as Director of Molecular Therapy. Welcome back, Tom.
Glad to join. Excited to have the conversation.
So, and the third guest is someone which I couldn't believe is the first time on the JNM podcast, at least with me. It's really hard to believe because he's one of the most renowned faces in nuclear medicine. It's Professor Jason Lewis. He is the Deputy Director also of the Sloan Kettering Institute. He has many titles, but he's also the Tau Chair in Oncology. Welcome, Jason.
Morning, Ken. Morning from a nice sunny New York City. You can see behind me.
You have by far the best background. There's no question about it. So you get the prize of the best background for 2025. Speaking of 2025, I want to kick it off by asking all three of you individually, what was the biggest highlight for 2025? Obviously, it would be nice to have some relationship with nuclear medicine. And maybe, Irene, we start with you.
A lot have been going on in the journal for the past year, but one paper I really liked was a recently published paper by the Medical University of Vienna, highlighting a new software using AI, which is called Puma, which makes it possible to overlap images acquired with different tracers, or even images obtained with the same tracer but at different time, so as to obtain multiplex images. And I think it's great. First, it serves multi-tracer imaging, which is a great of great use in our specialty, and it's free. So I really think it's a great resource for our field and a wonderful gift for the community.
Thank you, Irene. What about you, Jason? What is one of the favorite things you have seen in 2025?
In terms of JNM, actually, the field as a whole, it's been one of the best years we've ever had. But in terms of JNM, apart from our own papers, of course, from our own lab, which I won't get into, I think it's just the breadth and depth of the work that's come out. There's been I'm going to pick two papers. There was one which came from France, the 3D Potium Frank Denard paper, when they did a phenomenal job of reporting a new chelate, PYTA. Normally people put out the chelate out there and they're like, oh, this is better than anything. But they did the most beautiful systematic study comparing that with all the other chelates currently for actinium. And it was just nice to see some solid basic science, some solid chemistry, estimated chemistry, and just a really nice systematic study. The other one is, I think, something we've been trying to wrangle and deal with for many years, and that's trying to image T-cell activation. T cells are one of the big interesting areas right now. And there's a paper that came out from Stanford with Michelle James and Israt Alam. It's in the, I'm not sure which issue was actually, but they used an antibody to look at T cell function. And it was an OX40 PET tracer. And I think that, T cells are struggling. I'm not an oncologist, but in terms of solid tumors rather than liquid tumors, I think that imaging could be the way to help resolve or sort out many of the issues they're having by seeing our T cells being activated in enough quantities to actually do what they're supposed to do. So I picked these as my favorites because we have chemistry, we have biology, but they're asking and answering really fundamentally important questions that we need to deal with in the field.
So we heard what are the exciting things from physics and chemistry, radiochemistry. I myself know more about clinical application. That's why I'm really curious to hear what Tom is going to mention.
2025 was just a year that And obviously, theranostics is coming to the forefront on it. And with the approval of the Fiji PSMA a couple of years ago, I think you're seeing clinical experience come to the forefront. Right? People who have now treated large cohorts of patients, and people are starting to understand how we are going to practice medicine in this new era, which is new drugs. And that's what we're seeing starting to come through on the side, right? Lots of papers about SPECT CT and PET imaging, how we use these modalities for response biomarkers, toxicity prediction along those lines. And I think it's not one paper per se, rather the entire grouping of research is going on to help us understand what is this going to look like, right? You have, all the big phase 3 trials end up in big journals, but JNM is where where the tire hits the road. We have to learn how to actually do this. And I think that's what's finally getting out into the literature is how we're going to use these things clinically and use the tools available to us, particularly SPECT CT, understanding how we're going to apply that and use that, I think is really interesting. And then soon, hopefully in the coming years, sort of converting into dosimetry. We talk a lot about dosimetry, but I don't think that's really yet gotten into the clinical practice. But this year to me was more about the clinical experience, the first wave of theranostics as these drugs become more widely available. And it's been a lot of fun to see how people are struggling with the clinical application.
Thank you, Tom. So actually, our editor-in-chief, Jonas Chen, would be really proud of us because I asked you guys what are the most exciting things in nuclear medicine in 2025, and you all come back actually with things which are addressed in JNM. However, as much as we love JNM, there's even nuclear medicine outside of JNM, even so it's difficult to imagine. That's why I want to ask you one more time the same round, same sequence of people. What was else super exciting in 2025 in nuclear medicine or outside of nuclear medicine? It doesn't have to be connected to the JNM. Irene, tell us something super exciting from the field of artificial intelligence.
The fact that many of the AI tools that are currently developed are now going into a clinical evaluation. So people are starting to see what difference it can make, in a clinical setting, which is great. We no longer need proofs of concept. We already have them. We know it might become extremely useful in our field, but we need real demonstration of the added value of leveraging these tools for routine applications, for making new discoveries. So that's what we need now. And I think it's coming. We start seeing a large evaluation of, AI-based segmentation tools, of tools that can assist in reporting, make reporting faster and less operator dependent. So I think it's great that we now have these evaluation available so that all these AI promising results can translate into the clinics.
Thank you. And Jason, from your point of view, what was super exciting in 2025?
Most exciting, I think, the identification of new targets that we can start hitting with radiopharmaceuticals and therapy would be one. I'd like to hear what Tom's thoughts are on my next comment, but more of our radiopharmaceuticals therapeutic coming first line. I think we're going to see a lot more of that rather than last-ditch attempt at the end of 4 or 5 other treatments. But honestly, I think as a field, I think that, we've been somewhat just siloed, this kind of weird little group of people aside that have our meeting. And, I've been doing radiopharmaceutical for 30 years, and I've never felt there'd be more interest in what we do ever. And I think the fact that we're headlining at AACR, Astro, ESMO, ASCO, all of these mainstream, big, huge med-oncology and radiation oncology meetings, and theranostics is up front and center. I think that for me is really exciting because our field and its adoption of it more widespread is going to rely on these kind of connections we have with these other groups. And it's just been really exciting to see that happen this year.
Jason, I'm so happy that you mentioned the bringing PSMA lines a bit, but also actually Lutathera into earlier lines. Now, I'm not going to comment. It's the guy who's going to comment on the clinical is Tom. So, Tom, the floor is open.
Yeah, so I agree with Jason. To me, clinically, the main trend in this year was going earlier, right? PSMA4, the results of PSMA addition, NETTER2, all of this moving radioligand therapy into earlier stages. And I think I'll even add to that is that, IITs like Lunar to me were really sort of eye-opening. The idea
that you can treat really low volume disease. And I'm not sure I totally understand the results of that trial, frankly, what to do with it. But it's really eye-opening on how we use PSMA radioligand therapy and other treatments in low volume disease in combination with other drugs. And that to me is the trend of where we're going right now. What do I think of it? I'm a little hesitant in certain patient populations to go too early, right? I'm not a believer of the C word here. And we do always have to keep in mind long-term toxicities. And, one of the big trends this year was something that hasn't made it into the literature, but was at NAMETS, which is the renal toxicity associated with lead SSTR target agents. And I think that type of discussion is going to rear its head in a way we may not want in the coming year. So, it's coming. But there's going to be a concern of some of these long-term toxicities that we need to be very cognizant about. I think the other aspect of sort of this year, and to sort of go on for what Jason said, is new targets. Now, I can't understand everything about it. So to me, new targets are first in human studies. And there was a lot of interesting stuff that came out this year. GPC3, CA9, the APC3 images, which haven't yet really made it into the articles yet. There's a lot of new targets that I think are addressing a question that I've always asked for a couple of years. Is there anything beyond the somatostatin receptor and PSMA, or are they unique? And it's looking to me in this last year, there's going to be other targets like it. And we're going to have at least a handful of targets that are going to be coming out. And the results, and some of those are published in JNM, right, are really interesting, exciting. Biodistribution looks great. And I'm really looking forward to the clinical translation to the therapies. So I think both coming earlier, although with a little bit of hesitation, I probably am more hesitant than the average person in the field. And then also, obviously, these new targets and new agents that are really, really showing promise in a way that I don't think I've seen recently.
I'm very happy you mentioned APC3. I think it's super exciting target, at least for me, came out of nowhere. And it's going to be published actually very soon, unfortunately not in JNM, but that's maybe also the price of success that nowadays with really exciting stuff, you can even target papers which have a higher impact factor than JNM. This being said, our field is much more than just articles and publications. I think time around Christmas is also a time where you sometimes have time to actually listen to a couple of talks you might have missed during the year. And I want to ask each of you, what is a talk you really was impressed? We all traveled to different conferences. What is a talk you really suggest that someone, when you have time, go back and listen in if it's available? Jason, what comes to your mind?
So many. I mean, one of the things I've been trying to do is go to meetings which don't necessarily have nuclear medicine or radiochemistry up front and giving the talks, but going and listening to completely out the field talks and see if that's kind of information or studies that we can bring into our field, whether it be at the basic science or the translation or the clinical level. And I only can't remember who was giving the talk, but I was listening to some of the talks at ASCO this year, and the profound difference that some of these drugs are making in the patient. And I think the thing that sticks out to me is the fact that there was, I wish I was trying to find the talk before the podcast, where somebody was like, well, we know this drug really, really works well, and we've had a really good improvement of survival. I just wish we had a better way to pick our patient population on the treatment arms. And I'm sat there thinking, well, we could do this. With an image to that target at the beginning, you could absolutely decide who goes on what arm and what down the other. So it's not really a specific talk in general. It's just like some of the concepts and ideas which are out there and coming through that I think that we as a field can help answer. And I wish I could find that talk, because I wanted that little snippet to then put on my next slide and my next talk saying, if only there was a way we could do this. I'm like, we have a way. You just need to learn that way and we can help you. So that's really, I guess, my comment on that.
Thank you, Jason. Irene, what was a talk you really find memorable in 2025?
Again, it's very difficult to select just one out of so many. I really enjoyed a debate that was held during the annual meeting in New Orleans about, so it's AI again, about the value of these, magic wand based on AI, which makes it possible to retrieve very nice looking images from very low count acquisitions. And the topic of the debate was, is that a trap or is that a revolution to reconstruct great images from low count images. So it was quite interesting, and basically the true answer is in between. It's helpful, but you still need the counts if you want to get a real good estimate of the biodistribution of your tracer. So you should not go too low in count, and you should not trust these AI tools too much. You need counts in nuclear medicine. Keep that in mind, please.
Thank you, Irene. And Tom?
The session at the annual meeting that I found super interesting and eye-opening, and Jason was one of the presenters, about how animal models predict biodistribution in humans. And to me, it was very eye-opening and impacted a lot of how I think about animal models, but also I think it's going to, that type of thought process that animal models don't really predict what we see in humans very well is going to impact the regulatory processes in the coming years. And I think that session to me was really eye-opening to me about the limitations and where, there is value to animal models, but I think we may overstate their value in certain settings. And so I thought that was really eye-opening to me. The second talk was at our Theranosys conference, the SMI Theranosys. A person I've never met before, Fred from Happy Body, gave a talk to me that was very interesting about the impact of molecular size on theranostics and trying to get at where's this sweet spot for how big a molecule should be to optimize biodistribution and target uptake, which I think is something we're all struggling with in the theranostics space. And it was really interesting to hear, some history about molecular size that I wasn't that familiar with. And I thought that was a really very interesting thought. And then another talk I saw that I really, really liked was a talk in a closed conference that, wasn't able to have other people go see, but by Ititia Bardia. He's a medical oncologist at UCLA. He gave a talk that I found super interesting, which was laying out different ways to target proteins on cell circuits, right? So we talked about radioligand therapies. Medical oncology has, T-cell engager, CAR Ts, all these different ways of doing it, and starting to lay this framework about how do we think about what's the best way to do that. And those are three central things that are going on in my mind that came out of some of these talks that I've seen over this last year. I really enjoyed those three sessions.
Super. Thank you, Tom. I take liberty to add one more thing. I found a talk by Ambrose Baer during ENM Plenary 4, where he explained us why nuclear medicine should be taken over by radiology. I found it not only content-wise super, super good, I also found it really entertaining and also telling us something that we, as happy as we are and as happy everything is, we also have certain things we need to work on in the future. And this being said, I also want to, having three associate editors of the Journal of Nuclear Medicine joining me today, I also want to look a little bit forward, right? What are breakthroughs you expect in 2026? And what do you think is going to drive nuclear medicine, but also the JNM in 2026? And for fairness, Tom, I go back to you. You were always the last right now. Now I want to give you the opportunity to have less time to think about it. Tom, you're first.
To me, I think the thing that I'm interested to watch in this coming year is the novel targets, sort of the phase 1-2 stage of drugs, and both in novel targets, but also novel radionuclides. So astatine, lead, terbium. Seeing, I feel like we have a 3-horse race here into the non-actinium radionuclides out there. How are we going to think about those three, the actinium, the astatine, the lead, and the terbium, getting more experience with that, getting more publications and understanding toxicities? How does dose rate impact outcomes? These really short half-life agents deposit the dose much more quickly, et cetera. So I'm really excited to see that coming out. And then the new targets we talked about before, right? This last year to me was exciting, right? Again, that's the first year where I feel like, hey, we do have things other than PSMA and somatostatin receptor. We're going to see more. I'm excited. I hate to say it. We're going to see the first small molecule DLL3 PET images soon. That will be in the next year. Maybe Jason's not that excited because he's very obviously excited about the antibody approach. But how can we target these lower expressing molecules on cell surfaces and getting some of those images to really see these targets we've now talked about for maybe 4 years, all these things. We're going to start seeing the first in human images from these very soon. And I think that to me is really exciting to see the first clinical translation of these targets that we've discussed for a number of years finally make it into humans.
Tom, thanks for putting Jason on the spot. Makes it easy for me to pick the next person. Jason.
So, I mean, to Tom's comment about the new targets is completely spot on. I don't think we've scratched the surface on what we can look at. Many targets have been completely thrown away by industry because they're not expressed enough. But I think as our field has shown that a target that's low expressed like DLL3 is super valid. And Tom, we're doing the antibodies, but if they have a small molecule or peptide, very happy to do that. I started off as a small molecule guy, then I became a peptide guy, then I became an antibody guy, and now I'm whatever works guy. And if a small molecule works, then so be it. Might be hard to beat the antibody, but it's all good. But I think that all Tom's comments about the isotopes and everything are really important. I think, oddly, one of the things I'm most excited about is finding out what isn't going to work. We're making a huge amount of promises on all the stuff we can do, and that it's going to cure this, and it's going to cure that, we get to do that. One of the things we need to do is learn from our mistakes, and more importantly, learn by things that don't work. So when it does get to the clinic, and we learn about toxicities, we learn about the dosimetry and stuff like that, these things don't often get published if they don't work. But I think that value in what we can learn from those things, that we design new things, is super important. So I'm really hopefully looking forward to people saying, you know what, we tried this, didn't work. And then those with the basic science level can go back and work out why it didn't work and see if we can find an alternative that will. And I think that that's what I'm kind of oddly looking forward to, is finding out some of those things which don't work so we can do better and improve upon it, because the targets are great. You know, maybe we just need to design a better platform to hit them.
Thank you, Jason. Nothing is as transformative as AI. So that's why I think it absolutely is perfect that now actually Irene is telling us that whatever we talked about is irrelevant because AI is going to change everything. Irene, tell me what's happening in 2026.
Yeah, my expectations is really that AI will assist in the clinical translation of, the great work that you are doing. We have to make things easier to perform in the clinics. We know that we have highly demanding, RPT protocols, especially if you want to do dosimetry. And I really trust that AI can make things easier, faster, so that, all the promises of nuclear medicine can now be translated in many centers and not be used only in very top-end clinical centers. And another expectation that I have is that there will be more harmonization work. I mean, we have to agree on how to measure quantitative biomarkers from our great images, how to interpret them. We have to harmonize practice so that oncologists cannot challenge us, or hematologists, they cannot challenge us, by telling us that we should first agree on how to do things, and then they will trust our, what we deliver to them. So I think these harmonization efforts are absolutely needed in our field. They have started. Many people have realized that we are a small community. To be strong, we have to work together and come with consensus decision, consensus interpretation rules. So I hope we will improve in that respect.
Irene, thank you. So first of all, thanks to all three of you. This was an experiment. I really enjoyed very much. This was really fun. I also have a little bit of homework to do. I know now a couple of things I've apparently missed, and I want to read up on this and also look up on this. Also, I have to say, none of you mentioned JNM podcast, which means we have to do better next year to potentially make it to the highlights of 2026. But overall, like I said, it's an experiment. I hope that all of you give feedback to us if something we should repeat or not. But overall, I'm not worried about our future. It's going to be super exciting what I heard from you. I share your excitement. And with this, all have hopeful, peaceful holidays. Have a good start of 2026. And stay tuned. JNM will continue to stay very active. Thank you very much.
Thanks for watching. To see and hear more from 2025, search SNMMI or JNM Podcasts to find us on social media and wherever you get your podcasts.